Hello everyone, my name is Aadi Desai and I was a CCIR Future Scholar student for the fall program in 2023. I did my research under the mentorship of Dr. Begum Tunker and Dr. Tanner Tunker from the University of Cambridge on cancer biology and its mechanisms. My paper focused on studying one of the most extensively studied genes, TP53, alongside its mutant protein. However, before we dive into the objectives of my paper, I would like to give you a brief sketch of P53's history that highlights the work that went into discovering the nature of cancer's most important gene. In the summer of 1979, three independent groups of researchers in London, Paris, New York, and New Jersey hit the jackpot for genetic discovery. The protein they discovered, clinging to the monkey infecting SV40 virus, much like an incessant toddler, was found to possess a molecular weight of 53 kilodaltons and was thus named P53. No one at the time could possibly imagine what P53 would amount to be. But now, we know that P53 plays a pivotal role in instigating cell suicide or apoptosis in the event the cell runs the risk of turning cancerous. Termed aptly as the guardian of the genome, P53 prevents the spread of tumors through daughter cells and thereby ensures the integrity of the genome. It is no surprise then that mutations in P53 are considered to be a prerequisite to cancer formation. And as we can see in this graph here, TP53 mutations are found to transpire at high rates in breast, blood, ovarian, and uterine cancers, amongst many others. TP53, the gene containing the P53 recipe, is a tumor suppressor gene. However, with knowledge on TP53 being scarce and no other facts to prove otherwise, researchers in 1980 quite erroneously termed the gene to be an oncogene, or in layman's terms, a cancer accelerator. This was largely supported by the fact that there was a high abundance or overexpression of P53 in cells that turned cancerous. This was largely supported by the fact that there was a high abundance, right? So at that time, it was quite standard to conclude that proteins overexpressed in cancer cells must be produced by oncogenes. And so P53 in the eyes of researchers was quite plainly an oncogene. However, this was not so in the case of one person. Arnold Levine, the Princeton-based scientist and one of the discoverers of P53. No matter what he and his team did, they could not reproduce the results of overexpression that other scientific groups had achieved with their P53 clones. However, when they borrowed the P53 clones from other groups, they were able to do so. Unknown to Levine and others at the time, they were able to reproduce the results because what the others had been working on was that of the mutant P53, while the ones they had was of the wild type P53, or the protein that nature had actually intended to be. Now, more than 40 years since its discovery have passed, and with many other conclusive experiments conducted by scientists like Bert Vogelstein, as you can see here, have now established P53's job to be correctly that of pressing on the brakes rather than the pedal in cancer cells. As scientists' curiosity in P53 rose, and an atmosphere of intense competition was being established, one anomaly still caught the eyes of scientists, that of the overexpression of this innocent-looking tumor suppressor in a majority of the tumor cells. As scientists continued to speculate, Donald Dittmer in 1993 demonstrated the presence of gain-of-function mutations in P53. Gain-of-function mutations in P53 have not only caused it to lose its tumor suppressive functions, but also acquire new oncogenic abilities. And these abilities help it grow tumors as well as sustain angiogenesis and modify metabolism. And so, staying true to our car analogy, now, not only does the car lose its brake function, but also acquires a second, equally powerful accelerator, which causes it to speed up uncontrollably in terms of cell division. That brings me to my research paper, titled A Bioinformatics Analysis of the Transcriptional Interactions of Mutant P53 with Other Oncogenes. My paper aimed to focus on the interactions of mutant p53 and try to understand the means it carries out to conduct its functions. So now that I've established my research paper title, chose to focus on an entire data-oriented approach. And as was elementary to my course, I focused on a bioinformatics approach, which involved the use of several data sets like GEPIA, CBioportal, DepMap, YOLCAN, String, GeneMania, and Tagnomai. P53 is one of the most least and the most understood gene in cancer. And what I found was that the P53 expression in BRCA was quite high. BRCA stands for breast cancer. Within breast cancer, there were within breast cancer, breast invasive ductal carcinomas had the highest expression of mutant P53 or altered P53, which was at 89.42%. Now, invasive ductal carcinomas are mainly triple-negative breast cancers, which are characterized by the absence of estrogen, progesterone, and human epidermal growth receptors, or basal-like tumors, which are characterized by their low prognosis and their high aggressiveness, which is once again suggestive of oncogenic mutant p53 behavior. Furthermore, I then focused on understanding the genes associated with p53. I drew a heat map with the help of YOLCAN, which is a user-friendly comprehensive database that focuses on using various omics data from several databases. What I found was that several genes, such as PSAT1, LCN2, MMP1, and S100A9, were quite overexpressed in breast cancer with TP53 mutant tumors. Now, for example, the PSAT1 gene provides energy and anabolic support to the cell. The LCN2 gene facilitates cell proliferation, invasion, migration, angiogenesis, and immune regulation in breast carcinoma. And the S100A9 gene contributes to tumor development and metastasis by interfering with the tumor's microenvironment. The S100A9 gene is seen to be possessing several p53 binding sites that accommodates DNA binding activity. It was also demonstrated that S100A9 is a p53 target gene that induces partly p53 dependent apoptosis. MMP1 is also regulated by p53, and its dysregulation seen to promote tumor metastasis. Also, the PSAT1 gene is also identified to be interacting with the p5372p protein, which is a single nucleotide polymorphism of the codon 72 variants of p53. p53 is a transcriptional factor, and it carries out its guardian of the genome functions by facilitating the transcription of several other genes. And so I figured that this was the same thing that mutant p53 did. On the basis of my data, I hypothesized that mutant p53, upon undergoing gain of function mutations, activates the transcription of several known oncogenes in a bid to promote tumorigenesis. This hypothesis was further backed by graphs from YOLCAN, which showed that the expression of several of these oncogenes was quite high in triple-negative breast cancer and HER2-positive breast cancer, in the sense that the statistical differences between TNBC, HER2-positive, and normal tissue in these genes was quite high. So, having dissected the problem and with a working hypothesis at hand, I then set out to find viable solutions. Till date, p53 remains undruggable in the sense that no drug type has been developed to curb the mutant gene's actions. This is because it does not possess the typical drug target features one may expect from a gene and lacks the required deep grooves and active pockets for small molecules and antibodies to occupy. However, many new strategies have appeared over the years, the most promising of them being PROTACs. I used a machine learning algorithm called Tagnomai to analyze the druggability of p53. And as can be seen here, PROTACs showed a high rate of success in mutant genes, mutant p53 for this case. And so going back a slide, let me introduce to you what PROTACs actually are. PROTACs are also known as proteolysis targeting chimeras. They are molecules synthesized to target and bind to proteins, inhibit their biological function, and induce their proteasomal degradation, therefore destroying the protein in its entirety. So, PROTACs appear to be quite a promising strategy for mutant p53 breast cancer. Now, these are still big claims with a long way to go before they can be presented as hard facts. A lot of experimentation will have to go into proving that these interactions do exist. But if indeed they do, their implications seem to be far reaching. Studying these pathways could, for one, provide a detailed and deeper understanding of the workings of P53, or mutant P53, provide insights into what makes triple negative breast cancer so aggressive and having such a low prognosis. And three, they may also set the stage for future biomarkers and therapeutic strategies that may inhibit these genes' functions. That brings me to the end of my presentation. Thank you for listening. I would also like to thank the CCIR or the Cambridge Center for International Research for the support, as well as my professors, Dr. Tanner Dunker and Dr. Ackman Dunker, for their guidance. Thank you.
Thank you, Aadi, for a very thorough and interesting presentation pointing in the direction of some really important developments in breast cancer and all creative and innovative use.